Really welcome to all of you who are attending this meeting. This is our fourth installment of special topics in cell isolation webinar series. Stu presented the second one in this series, and this is a follow-up to his earlier presentation on adipose tissue. I want to welcome Stu Williams for his second tour of duty, so to speak, for this webinar series. And he's going to talk about the characterization and therapeutic use of adipose-derived cells, some inconvenient truths, as you see on the slide that is on the screen. Stu, I think, likely has more experience in the study of adipose tissue from a research perspective than anyone else in the world at this time. He started actually during his PhD training at the University of Delaware, followed by postdoc at Yale, and you can see the school icons. And basically went to Thomas Jefferson, had faculty positions at Thomas Jefferson, University of Arizona in Louisville. He's now retired from the University of Louisville. I really value his expertise in this topic, which is the reason why I've asked him again to expand on and have a different perspective on some other aspects of adipose. So without further ado, I will turn it over to Stu.
Okay, thanks, Bob, and thanks to VitaSite for putting these on. They're incredibly educational to me. As Bob mentioned, that's the title of my talk. These are all my lineage. I actually started in third grade. We were just talking about that, studying the microcirculation of the chick egg as it developed, and have continued to study the microcirculation and its relationship to fat throughout my career. This is being recorded, and a lot of people jump onto these presentations well after the live presentation is over. So I mention that because I've given my email. You can leave comments on the YouTube, and we will take a look at those comments and try to respond to them. Or you can get in touch with me directly at cvregen@gmail.com. That's my email address. All righty. So financial disclosures, I have quite a few issued U.S. patents and foreign patents in the field of regenerative medicine, as well as a number of patents in the area of adipose-derived cells, and I've been previously a consultant to numerous biotech companies. I started with this term, inconvenient truths. That comes from an Al Gore documentary from 2006, where he was talking about the inconvenient truth, the effect of CO2. There are a lot of inconvenient untruths also. Began in 1987 with a Valvoline commercial, where it was stated motor oil is motor oil, and pretty quickly it was recognized that not all motor oils are the same. Valvoline sold a lot of oil based upon that, but there are at least four untruths that come out of this field of fat-derived cells. And here are the four big ones. Fat is fat. FDA basically lives on this untruth all the time, that with a very simple definition of what fat is a structural tissue, and not all fat is the same. Not all SVF is the same. Endothelium is incorrect, and especially for VitaSite and those of us dealing with enzymatic digestion, collagenase is an untruth. There are so many different forms of collagenase that we use for tissue dissociation, and that was a big focus of my previous presentation. What I want to do in this presentation is focus on a couple of things. One, the characterization of these cells that we find in adipose tissue, how they formed, how we isolate them, and what we call them. And I'll spend a little bit of time talking about the influence of this word stem cells and Arnie Kaplan's infamous paper from 1991, where it really drove us to redefine cells coming out of lots of enzymatic digestions. And I will, in that part of it, to provide you with what I think we should call these cells isolated from fat using a variety of different methods. It all started as far as the assessment of truly what is in fat and what the specific cells that create adipocytes by work by a guy by the name of Fleming, 1870. It was all in German. I remember this paper well because when I was doing my PhD, I had to pick a couple of foreign language papers and translate them myself. We didn't have Google at that point it was 1978. So this is one of the ones that I had to translate and hand it over to a professor who was German to see if I did a good job translating it. But the most important thing that I realized here is his statement that the pre-adipose cell that is in fat, you can't really define it as one specific cell. There are multiple cells in fat that basically participate in the formation of new fat. That's 1871. And then a very famous paper that came out from the Clarks at the University of Pennsylvania, Elliott and Eleanor Clark. They were anatomists, and a lot of this initial work was done by anatomists. And the most critical thing that they found studying the development of fat morphologically, anatomically, was new fat cells did not form right at the point where angiogenesis took place, but at the point where the new blood vessels had formed, undergone some level of differentiation, maturation. And it was from those endothelial cells that fat was derived. And then they also noted that if inflammation took place, you could see a reduction in the amount of fat. And they should have patented it at the University of Pennsylvania because this is part of the mechanism of Ozempic now, GLP-1. But their anatomical view really did set the stage that endothelial cells were the critical progenitor cell for all the cells that occur in fat. And then a paper by Russ Barnett. Russ was the head of anatomy at Yale School of Medicine, and I got to work under him when I was at Yale in the late 70s, and he gave me a copy of a paper that he wrote for the Duell Conference, sometimes called the Duell Conference. Howard Duell was a biochemist who studied lipids, and they have a conference held in Southern California for the most part all the time, every year. They still hold it today. And it was at this proceeding in 1962 that Russ Barnett wrote one of the definitive papers on what adipose tissue looks like, what's in it. And I enjoyed my conversations with him as we went through that the embryology of these cells and fat truly comes from the vasculature. They are the progenitor cells that exist there. And another huge paper was 1978. Faust and his colleagues at Rockefeller University really changed this whole field of our understanding of where new adipocytes come from. Before 1978, when I took physiology as a course, I learned that you're born with a certain number of adipocytes, and then those adipocytes get bigger or smaller depending upon your diet, genetics, et cetera. It was 78 when Faust said, no, 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 no. New fat cells come from progenitor fat cells or progenitor cells in your fat. So we now have to start looking very actively for these progenitor cells and answer the question, where do these new adipocytes come from? It changed my lectures in physiology when I started teaching physiology at Jefferson, but also was intriguing to me because it's now suggesting that there are stem cells in fat that are critical for obesity. It leads to this, which is one of the first models that I'll present. And as we all know, models are great until they're not great. But this is the model that I have developed over time based upon the hundreds of papers that I've read by anatomists and others, where fat comes from. It starts with mesodermal cells. A blood island is formed. That blood island can form the bone marrow, hematopoietic stem cells. It can also, the blood island can go on to form the microcirculation. And then critical to the discussion today, as new blood vessels are formed, there are tip cells which basically migrate through the connective tissue. And the cells behind the tip cells that are undergoing maturation, these are the source of cells. They're endothelial cells that form vascular smooth muscle, the new circulation, endothelial cells to form the adipose microcirculation, hemogenic endothelium. These are the endothelium that can directly differentiate into hematopoietic cells, hematopoietic stem cells, pre-adipocytes, lymphatic endothelium. The only cells that are not coming from these endothelial cells are the sympathetic nerve cells, the sympathetic innervation, and then there are circulating cells that will diapedese into the fat over time. They will increase or decrease the total number of cells. But when I look at the literature, and this is the first inconvenient truth that I will throw out there and open up for discussion later, as well as on YouTube and other places, it is the endothelial cell which is the progenitor cell to all cells and fat except these nerve cells and the circulating cells that diapedese in. So all cells and fat are derived from endothelial cells is my first inconvenient truth, to use that term that Al Gore has used over the years. It has led us to a characterization of the cells, isolated fat from fat, utilizing enzymatic digestion. And I will make that distinction in a few minutes, enzymatic versus non-enzymatic cells that are isolated. And I showed this slide in my first presentation. I'll show it again. In the literature, for example, if you look to see what percentage of cells in the SVF, the primary enzymatic isolation from fat, what percentage of cells are endothelium, you will go from 0% endothelium in fat to 100% endothelium in fat. So all the cells in fat are endothelial cells, and lots of percentages in between. One of the first times I gave a presentation on isolating endothelial cells from fat, I had a number of surgeons who came up to me to correct me, saying that fat is not vascularized, and their proof was, they said every time they cut into fat, it doesn't bleed. So if it doesn't bleed, there are no blood vessels, so there are no endothelial cells in fat. So the only way for me to get out of that conversation was to say that they were incredibly skilled surgeons, so they could cut around the blood vessels and would not see any bleeding in that fat. Fat is highly endothelialized, and it has many other cells that we're going to talk about. The fibroblasts, pre-adipocytes, parasites we'll talk about also, macrophages, leukocytes, and the importance of these cells in a number of different, not only basic science studies, but therapeutic applications. I have two what the medical students called "what the hell" slides. Don't turn away totally from these. This first one is basically from the Marty Rodbell 1964 paper, Isolation of Cells, how these cells were utilized by different key investigators and then people who got into those particular areas to study different functions of the cells from fat. The adipocytes themselves were studied mostly by the lipid biochemists to try to understand how the adipocyte stores lipids, how the adipocyte metabolizes, and Rodbell got the Nobel Prize for his understanding that these cells had membrane-bound transducers. Endothelial cells were studied for their function, especially the function of capillaries, and were also used since 1972 in regenerative medicine studies and then on into transplantation. Microvascular fragments, the work that Roger Wagner started. 91, all of a sudden we changed the name of these cells to mesenchymal stem cells because of Arnie Kaplan. In 80, the Bjorn Torp, who's the major investigator for these types of studies, began to call these cells from the SVF adipose stem cells and showed that they could form all sorts of different types of cells. The Faust paper in 78, all of a sudden we started looking for pre-adipocytes. And here are all of the different, I call functional studies that were done: obesity, regenerative medicine, transplantation, and signal transduction of the cells, as well as a variety of other different diseases that we began to study using this. These little yellow spots are interesting from the standpoint of how information on all of these different studies was disseminated. Eugene Garfield out of Philadelphia, started Current Contents, the Science Citation Index, in 63. As an author, you had to provide your own index of the papers, and then he would send out a paperback booklet every week that had all the papers that were published. Incredibly important for those of us who were trying to figure out which papers to read. You didn't have to go to the library to try to figure this all out. You could do it straight from Garfield's. It was self-indexing that caused some issues. Meta-analysis 77, EndNote came out in 89. And then a big one that Al Gore did is he got the federal government to pay for Medline so everybody could use it for free. This opened up information on scientific publications to everybody. And then Google Labs started in 2002. And if you haven't seen it, take a look at Notebook LM, which is an AI platform where you can put PDFs of scientific papers in and ask specific questions. I'm having a lot of fun utilizing this and asking simple questions like, where do the stem cells and fat come from? And depending upon the papers you put in, you'll get pretty much a uniform answer that it's endothelial cells. And then this one is a even more complex slide that what I'll say is I am nearly completing this. I'll put it up on my website. Bob may put it up on his website so you can go and truly see all of the publications that started with Rod Bell's cell isolation method and led to the in vitro and in vivo differentiation of fat-derived cells into a variety of different cells. I'm making this available especially to attorneys involved with intellectual property on where all these different cells can come from the stromal vascular fraction. And most of it dates back to the early 80s when those discussions were took place. All right, we're going to go through this one somewhat quickly. Stem cells. This terminology really came into place to help what I'll call a simplistic view of what is in fat, what is in bone marrow, and what cell is the most important for derivation of other types of cells for therapeutics. I call this an inconvenient truth that the field of stem cell biology has been hampered by this utilization of this term stem cells. Why? In NIH and so many different funding agencies, molecular level mechanisms rather than outcomes were the focus of studies. It uses a reductionist approach. There is a total focus on purified cells. When we start talking about stem cells in these different tissues, everybody wants to isolate a purified cell that could then proliferate. And then all of a sudden they home in on a set of CD markers, and that is the law of the land. It's purely reductionist, simplistic approach, a focus on what are called hierarchical stem cell models and what is known as heuristic bias. Societies all of a sudden use their hierarchical structure to create policy generating subcommittees, and that became the law of the land. And I think this has hampered this field incredibly. One of the major things is if you focus on one cell population that has stable CD markers, you will lose sight of the fact that most pluripotent stem cells and most pluripotent cells in any tissue, including fat, are actively cycling and actively changing their phenotype. So if you lock in on a set of CD markers, you're not going to take advantage of the entire population of cells. So Arnie's paper, 91, we lost Arnie last year. He passed away. He set us on a path that's critically important, that there are certain forms of stem cells and medicinal cells in all tissues, including fat. But it was overblown, I feel, that if you're going to study any sort of therapeutic cell out of a tissue, it's got to be a stem cell. And then the ISCT got involved, and they said the stem cells or stromal cells have to be fibroblast-like, and they have to be plastic adherent. And that has several problems in our definition of stem cells. And then they used a reductionist hierarchical approach and said all MSCs have to express or not express these markers. That has hampered us also in this field. Arnie came along and said all MSCs are parasites. And that may be true if we define parasites as an endothelial cell with a slight differentiation, but true parasites, as I'll show you in a moment, are really essentially devoid in adipose tissue. It's not the parasites. This picture is probably one of the most overused examples of parasites surrounding the microcirculation. I've seen it in many presentations. It doesn't appear as much in publications, but still people have it in presentations of the parasites. It comes from Roger Wagner's and Fred Hostler's 1992 paper of parasites enveloping the endothelium of the swim bladder of the American eel. It has nothing to do with the cells that wrap themselves around the endothelial cells of fat, which leads us to another inconvenient truth. Parasites are relatively rare in adipose tissue. They are not the source of cells for, they're not a pluripotent cell that you can really find. And I just asked people to stop using Roger Wagner's figure 7 from that original paper. In 2010, Arnie changed the name, wanted to change the name of MSCs to medicinal signaling cells, beginning to help us develop the idea that these cells have a paracrine function, and the cells may actually give off these little things called exosomes, vesicles that hold all sorts of material. 2019, the ISC got back into this and began to say, okay, the term acronym, it should be used for all tissues. So if it's a adipose MSC, you have to put an A MSC after it. In 2021, a couple of interesting editorials came out. One, Scott Rodeo from the Hospital for Special Surgery, orthopedic surgeon, fantastic surgeon, and a fantastic stem cell biologist. He really took Arnie on. They were, Arnie passed now, but they had a lot of fun with this discussion. And what Scott basically said was mesenchymal stem cells have really push the field back in orthopedic surgery and many other fields. He states it conjures up the image of a cell that can support the formation of a variety of new tissues after transplantation. And Artie admitted about a month later that Scott was absolutely right, and he apologized to the orthopedic community for calling the cells mesenchymal stem cells. But this term continues to live on today. This is the latest from 2023 now. In biobanking standards, MSCs, I really want to just leave you with this, an inconvenient truth. The use of the acronym MSCs and its various definitions and forms and functions basically confine it to the discussion of bone marrow cells and leave the adipose cells alone. I'll let Peter Quisenberry fight it out in bone marrow, and he's doing a pretty good job. That the true pluripotent stem cell in bone marrow may be quite different than these MSCs. But in adipose tissue, it's time to stop using that term, and we need a new set of terms that I'm going to present now. A couple of other inconvenient truths. A fibroblast is a phenotype and not a cell type. All CD markers reported specific for fibroblasts are also found on endothelial cells, depending upon where the endothelial cell is cycling. So to try to define the fibroblast as a cell is incorrect. So where do we stand terminology to describe these cells from fat? All started with Marty Rodbell, enzymatic isolation of cells. and he called these cells in the pellet stromal vascular cells. That's where it all began. It was fat, you mince it, treat it with an enzyme under agitation. The choice of that enzyme is important. He used a Worthington class 1 collagenase. That is the model enzyme mixture that we should still focus on today. It's centrifugation, no filtration, and then the final product is fat cells that he studied and the stromal vascular cells that he left up to the rest of us to study. These are the three basic methods that developed through 1978 for the isolation of cells from fat. Rodbell's studies that resulted in the stromal vascular fraction. Wagner modified it by putting a 200 micron filter in, and he called the cells that were trapped capillaries. You'll see the CD markers that I've put for you here. These are CD29 positive, CD31 positive, and the stromal vascular fraction is CD45 positive because it still has formed elements. In Wagner's method, the CD45 cells fall through. In Bjorntorp's, and Per Bjorntorp did some of the most incredible studies of pre-adipocytes and adipocyte development and his studies of obesity. This is his method. Mincing enzyme, same one that Rodbell used, agitation, and then he put the isolate through two filters, a 250 micron and a 25 micron. All the endothelial cells came out, so it was CD31 negative. Some were CD29 positive, and many of the cells were CD45 positive. There is a whole group of investigators that use this method. They are adipose stromal fraction cells that Bjorntorp developed. Many use Wagner's method. These are called microvascular fragments, and then Rodbell's method for the stromal vascular fraction. And then very quickly, the non-enzymatic method was developed by Carrel in his explant studies in 1923. And it's sort of fun. We have two of the four people here got Nobel Prizes for their research in studies. So Carrel developed the method for mincing fat, and he did all sorts of tissues, agitating it, centrifuging it, and then he didn't call it the stromal fraction. He called it an explant population, but these are stromal fraction cells. So here are the four methods, and I want to also put somewhat to rest that if you do a non-enzymatic method for the isolation of cells from fat, you will not get the vascular fraction. And if you look at the bottom left here, this is a scanning EM of liposuction fat, and it is an endothelial cell coursing across the top of these fat cells. It is entrapped in matrix. Only enzymes will digest this matrix. So investigators who use stromal vascular fraction to define the cells that come out not using an enzyme, so non-enzymatic methods, there are very few endothelial cells in that final fraction. Only the enzyme, and this is an enzymatic digestion taking place, will the cells be released from their extracellular matrix. Presented this before, and just to remind everybody, from 64 through 2003, this pellet was defined as all sorts of different things. I called it microvascular endothelial cells. Halverson talked about adipose tissue derived stromal cells. Zook talked about PLA, process light. It's all the same cell population if it's enzymatic and there's no filtration that takes place. So an inconvenient truth. After reviewing thousands of publications reporting the isolation of fat-derived cells, I have to conclude that there are not sufficient methods stated to allow anybody to duplicate other people's results. We need standardization of this terminology and methodology so people can take a look at your methods and say, oh, you're using this method with this modification. I recommend the following terminology come out of these isolation techniques. If you use enzymatic digestion with collagenase and protease greater than 20 minutes and a filter that's greater than 200 microns, it should be called the adipose stromal vascular fraction, and you should reference Rodbell in your publication. Any modification of Rodbell, adding filters, etc., you can point out, but this is Rodbell's method for SVF isolation. Bjorn Torp gave us collagenase and protease, but greater than 20 minutes, but he used a filter that was smaller than 200 microns, 25 microns. This is adipose stromal cells that he's isolating. Wagner captures the cells with a smaller, shorter collagenase digestion. That's adipose microvascular fragments. And then non-enzymatic tissue disruption. These are adipose stromal cells. This is not the adipose stromal vascular fraction. So I'm restating this in written form. If you're using one of these 4 investigators' methods, Carell, Wagner, Bjorntorp, Rodbell, use that as the primary reference and then give your methodological changes and show all of your methodological changes. What are the procedures? What filters are you using? And if you send it off for fax, find out whether people are putting a 20 micron filter in the system before they put it in the fax instrument. The cells that are analyzed by fax sometimes have nothing to do with what the primary isolate is. And stop using this term that the SVF cells are isolated according to our previously published methods. I have found so many papers that when you go back in the literature, you will look for the previously published methods and you won't even find them. It's time that we get a little bit clearer on the methods. These are all the different changes that have taken place, and I'm getting ready to complete all of this with a couple of review papers I'm going to write, and I'll talk about all of these different modifications and ways of trying to standardize these methods from Rodbell's original way to what we're doing today, and all the different filters, the enzymes that have been used, this filter that's used before fax is done. So that's the terminology. I hope we can begin to move towards, get away from just, you do a digestion of fat and what you get is stem cells. So many patients are told, well, we're putting stem cells back in. It's truly not the case, and there are differences in these different methods. So a little bit to finish up now on some of the regulatory, to bring everybody up to date where regulations are. The FDA regulates fat-derived cells according to the 1271 guidelines. They're considered HCTPs, so human cells or tissues. We have a real problem here because the FDA uses a definition of fat as a structural tissue that I believe came either out of a Google search that someone at FDA used, or it was from a histology textbook from the 1980s. We have evolved significantly from this definition of what adipose tissue is. There are multiple types of adipose tissue, and I think this paper by Kershaw and Flyer, as well as Russ Barnett's paper from 1962, states it pretty clearly with compelling data. Fat is adipose tissue, is an endocrine organ, and it should be regulated as an organ. It is an organ, it's vascularized. The FDA does not regulate vascularized organs. The FDA doesn't regulate blood vessels recovered with organs. This came from HRSA and FDA getting together and recognizing that people, when they isolate a kidney, will bring blood vessels with it, or they'll pull saphenous vein. They shouldn't be in the field of regulating those blood vessels. Well, microvascular fragments from fat are blood vessels, so they shouldn't be regulating microvascularized fat. We'll see how that flies with FDA. FDA's risk-based approach for HCTPs, lower risk 361, higher risk 351. So we'd like to be in the 361 field, but for fat, the FDA is pushing us into a highly regulated process to get fat-derived cells. I wanted to show you this, and I've worked in the field of fat for many years. I've also been involved in autologous islet transplantation, where the islets are isolated from pancreas. If you take fat out of someone or you take pancreas out of someone and you process it, you will see commonality to the mincing process, the collagenase digestion, the centrifugation, the pellet is washed, the product is released by quality assurance based upon purely cell number. And in pancreas, these islets are given back to the patient. It can be done remotely. We had, I helped set up a remote laboratory in New Albany, Indiana, where the pancreas could be flown in, processed one day, and then the islets flown back the second day. And 60% of this islet population is contaminated. It's with fat. Now we have to ask the question, how's it regulated? The FDA regulates this process as a 361 product. It is, they don't regulate it. They deem it as a 361 product, so it's not regulated by them. They will come and inspect your laboratory where you're doing the isolation, but it is a autologous, minimally manipulated, 60% of the time contaminated product that goes back into these patients. And it's reimbursed by most third-party payers, not reimbursed by Medicare at this point. Fat isolated according to the exact same methodology, but since fat is considered a structural tissue, it is going to be regulated by FDA through the IND process. I do want to point out that in SVF, if you take the SVF and you put it on an approved vascular device, such as a vascular graft, it's regulated as an IDE and not an IND. If you take the SVF and you isolate it according to an automated system, two examples are the Tissue Genesis Isolator system and the GID SVF2 system. All of their clinical trials are being performed under IDEs. And then adipose microvascular fragments themselves should be regulated by FDA through the blood vessel exemption. So I encourage any of you who are in clinical trials who might want to try that out with the FDA, give it a try. And then there are so many clinical trials that have completed phase 1 for SVF. The FDA Right to Try Act, once you've gotten through the phase 1 trial and have data from that, the FDA will allow a right to try in certain patients, and their definitions of those patients, terminally ill or they've tried every other procedure out there, and that is being utilized. Here's my another oh what the hell slide, but it's where I've taken everything, and again, I'm going to have this up website as well as in a publication where you can go and look as far as SVF or any sort of adipose-derived cell. You can see all the indications that are under evaluation. You will be able to see the mechanisms of action in papers as well as patents and clinical trials that these cells are being reported to have neurogenesis, hematopoiesis. You can take the SVF population and in animals, inject it into an irradiated animal, and hematopoiesis will take place. That's one example of many examples. And then this definition of what the therapeutic cells are. Hopefully, we'll get some discussion, comments, or you agree with me. These are not adipose mesenchymal stem cells. They're either the vascular fraction, stromal fraction, or they're microvascular fragments. It takes a village, all of these cells, to get all of these mechanisms operating in order to treat some of these complex diseases. When it comes to my use over the years, the first one I tried was isolating the endothelial cell population from fat, putting it on a graft and planting into patients. We had to decide where to do it, operating room or set up clean rooms. I visited UPS in the mid 80s to see whether we could set up a clean room there and process all the fat in the world in a biofoundry there. And then you got to determine what methods you're going to use. And our first method was we took the whole thing into the operating room in 88 and did the processing according to a slight modification of the Rod Bell method and put it into the patients and this patient and saw in vivo differentiation of these cells in this particular patient. Last couple of things. We've got some issues as far as all the different regulations that you need to follow for the isolation of many of these different cell types, including adipose SVF. One is quality system regulations, the 21 CFR 820. I'm going to make an inconvenient truth after, and I was a director of quality systems, quality assurance for one of the companies that I consulted with. And I've come to this conclusion. Quality systems provide an excellent means to assess and control the process. However, if you focus on this process only without evaluating outcomes, the final product eventually will not work. And this is a problem of dealing with quality assurance. We've got to make that sure that outcomes are evaluated. And when it comes to the ISCT efforts, if you force everybody to define these cells by a specific set of CD markers, positive or negative, their ability to adhere to blank polystyrene, it is going to become problematic because you may end up with a cell that has no therapeutic value or limited therapeutic value for the clinical applications that you have in mind. And then we have to talk potency real quickly. How do we determine how many cells? I'll leave it with Tony Dorsett. He said it the best after he was treated with stromal vascular cells back in Mexico. He said, I felt good, but then it was gone. But I guess you've got to get so many million, zillion, trillion cells in your body over a period of time. So this is current 2015, 10 years ago. If you try to look at how many cells do we get out of fat, it's interesting the variability in the number of cells you get out of fat, including in 2023, 2.2 times 10 to the 7th cells using a non-enzymatic method per gram of fat utilizing this particular method. We still need standardization of methods for how many cells we can isolate from fat, and we still need to understand and explain to FDA fat is not fat. Fat from different sources or different disease conditions can have a larger number population of one cell versus the other, which makes it difficult for the regulatory definition of what is in your cell product. Characterizing SVF is very difficult. Characterizing the ISCT stem cell is relatively simple. It just has to have these CD markers and not these other CD markers, but that ISCT mesenchymal stem cell may not have any therapeutic value. So one truth, the only place that SVF dosing has been worked out is for treatment of the internal surface of vascular grafts, where the optimal dose is 2 times 10 to the 5th cells. For all other applications, start with 1 million cells. That's where everybody starts and work up for there. We've got to do better at understanding it. And then I put this in, you can look at it later. You've got to understand reimbursement also. One of the big things that kills many, many companies is when they do get FDA clearance for their product, and then they go to try to get reimbursement. And then you go and try to get reimbursement from Medicare, and then you talk to hospital administrators, and then you find out about coding and billing. It's sometimes the darkest time for industries when they get FDA clearance. And if you want the best example of that, Advanced Tissue Sciences Dermagraft. When FDA clearance came, then they actually had to go back and start looking at what the costs were of taking. And I think we got, Kevin, a few others who remember those days very well. So FDA's got to change how they define an organ. We need to utilize only 3 terms to talk about the uncultured cell products from fat, SVF, stromal cells, or fragments. All investigators who are publishing patents, whatever, they should cite one of these 4 original methods. And clinicians investigating the use of these cells, at a minimum, they should follow good tissue practices. I'm going to finish there and open up for any discussions. Here are all the people that I've worked with over the years at all of these different institutions. And I'll also end on this slide. There's my email. And then that's the website. I'll be posting some of these slides with a lot more what I'll call the references that defend some of these inconvenient truths that I've talked about. That's all I got. I open it up for any discussion.
To me, just the general comment was a fire hose of information. I mean, I'm overwhelmed. And I think the thing that to me really stood out is how paradigms really are damaging. And trying to codify things the way one person sees it hinders science. And that was very nicely brought out.
Yeah, these, and I don't want to be too upset or pejorative, negative about, like ISCT. So they form a committee, and those committees change as far as the people come in. Maybe I could talk about it this way. The NIH uses study section committees, and all of us have dealt with that, whether it's SBIRs or other NIH R01 grants, et cetera. And those committees are formed, and that's somewhat of this hierarchical structure. Who determines who's on those committees, and then how the committee gets information to make a decision on whether, and you're not allowed to, for those of you who served on study section, you're not allowed to use what we call the F word there, the funding word. Should it be funded or not? What you're supposed to do is just talk about the scientific validity of the studies that are being produced, which is one issue with an SBIR, because you really want to know whether the product works or not whether it's the mechanisms are fully worked out as far as what the cell does. But this hierarchical, that it is a structure, and the study section has to make a decision, and it's based upon information that the study section brings to that event. And often the reductionist theory comes into the study sections,
Right.
or the reductionist theory comes into a scientific advisory board for a company, where when you're trying to explain why you're for or against the technology, you'll oversimplify your explanation. That's the reductionist problem that we have. You'll use the heuristic bias that exists in this field so incredibly, where you'll basically try to simplify your answer, and on the spur of the moment come up with a simple analogy. And I'm one of the worst people at doing that. I love to use analogies, but often those analogies just are too simplistic. The SVF works most likely because you inefficiently wash it and there are exosomes in it. You inefficiently wash it and there are other proteins that come along with it. And it is such a mixed population of cells that, just like in the body, all those cells work in concert in order to get you to your therapeutic application. I think if there's one takeaway from this, it's if we follow quality on process, you'll end up with a product that probably will not work in the end, because you drive in quality systems, you drive to make it the best process that is out there with no concerns with outcomes. And if we have to fully define the characteristics of this cell population, you'll end up with a cell that is defined by stem cell vernacular terminology, but it probably won't have therapeutic value.
Right. So on the chat, Stu, I mean, Kevin mentioned, Stu, great talk. What are your thoughts about which preparation method might yield the best immunomodulating secretome slash EV population? And is it likely that an SVF-based EV product could meet the current regulatory requirements for process control and lot-to-lot reproducibility?
So, great question, Kevin. So, one of the things that we've done, a couple of things to mention here. I worked in Louisville with the hand transplant program there. That's where all the first hand transplants were done in the world, and sat down with the transplant surgeons there and talked about the immunogenicity. It's a huge surface area of endothelium when you do a hand transplant, and talked about the immunoregulatory effects of stem cell populations, adipose SVF, and I just gave them all the different possibilities there. So ended up getting a clearance to try this out on a couple of hand transplant patients, and it showed that in a dual transplant patient, when we treated one hand with the SVF, the amount of immunoregulatory drugs that were necessary could be reduced. So it's a single patient, as always, but it helps us begin to develop this idea that there's something in this SVF population. We've done a series of animal studies also on this, we and others, that there is something in this SVF population. So now the question is, could you take vesicles, exosomes, the vesicles from the SVF and get the same result? Probably could. It would be better, Kevin, for everyone to get that through the regulatory process, because it's much easier to define your exosomes than it is to define this SVF population that's so complex. But my general belief is, unproven at this point, that immunomodulation that takes place most likely is going to come from all the different cells that are found in the SVF that are having an effect on immune recognition of the transplanted tissue that goes in. And in some of these cases, we perfuse the SVF into the vasculature, arterial perfusion in, so it takes up residence in those vessels. So it's now the patient's own endothelial cells being injected into a donor hand. That's been tried. A group in Taiwan has tried it for heart transplants. There's a group out of Allegheny Hospital that tried the SVF, and anecdotally did it in some animal studies, but didn't move beyond that. So didn't get into the human studies. I think the Taiwan group has done it. There's something in there, and we just don't know what it is.
So please, if people want to just use their unmute themselves and ask questions, please do.
Stu, this is Kevin. Thanks a lot for that info. You know, I think this is the big challenge in developing complex biologics, and it has been since you and I and the rest of the groups started it back in the early 2000s, is trying to fit our paradigm into the traditional single molecule pharma model, right?
Right.
So, you know, I think the organizations, as we continue to move along, need to come up with a concerted effort to continue to educate the regulatory agencies. Perhaps now, like no other time, the administrations are such that people may be more amenable to changing the rules than before. So it'll be interesting 4 years to see if we gain any traction for these sorts of things. But I just wanted to thank you for your continued leadership in the field and appreciate all you've done over the years.
Thanks, Kevin. So one of the things I'd recommend you do is send that slide to FDA where I'm calling them out on saying that fat is a organ, but and not the way they define it. But the only thing I ask you to do is tell them that Bob McCarthy came up with that slide.
Will do.
Yeah. Let's place blame very, very carefully.
Very carefully. It all began with this webinar. Any other questions people have from the unmute yourself? I can comment on a question. The other thing that impressed me is this whole open science issue. And I actually wrote a blog post on open science because, as many people know, half of the research studies, even when they try to repeat them and go to the original investigators, don't work. They can't be repeated.
Yeah.
And a lot of it, as you correctly pointed out, is because methods don't get their just due. They're just not described. So just FYI for anyone who's interested in it, if you're not familiar, there's a website called protocols.io, protocols.io, and just is a general a comment, that it's a very flexible platform, and if you go to their website, they're going to have an introduction to the protocols.io platform. And now a lot of journals are requiring or urging people to put their protocols on protocols.io and cite them in their publications because it is a citable publication. We are planning, just FYI, we have a much simpler collagen degradation activity assay, and I'm in the process of getting an intern to start working on that project. We've done it here. It's a very, very simple assay. I'm sure it will be accepted as a standard assay, but those are the types of things, and you correctly pointed out that if you're doing a method that's already been described and you're doing it close by, you should be citing the method and making the appropriate, if you're doing modifications, using the appropriate reference and citing the appropriate modifications you made. So I think it's an excellent point.
Yeah, thanks. But one of the things to spend a moment on, in the early days of isolating these cells and endothelial cells, I'd get a lot of call from investigators who wanted to repeat what I did. And they said, well, I repeated it, and I didn't get the cells that you got out. As a matter of fact, the fat didn't digest at all. And the first thing that I'd always ask them was, well, what were your conditions? Which collagenase did you use? And that was the collagenase is collagenase untruth that would always come up. And they would always ask, well, what collagenase did you use? So I would give them the manufacturer and the lot number. And they would call me back about 3 days later and say, well, I called up the manufacturer, but they said, Stu, bought out the whole lot, because the amount of time and effort and cost of doing lot selection,
Yeah.
and I found a good lot, I'd say, well, hell, we're going to buy this whole lot and then use it in the next 5 years. So I would send out some of my collagenase that I had for them to do testing so they could avoid the lot evaluation, and then they would send it off. I think what you're doing with VitaCite and being able to now use these to find what you did with the pancreas was incredibly important, and we need to bring that into fat and all these other cells where we're using purified enzymes in order to do the isolation consistently, and I don't have to give up my stocks of collagenase, which I've got lots frozen off in different places. So I got it from the beginning. Oh my golly.
Well, your experience also shows you, to me, because we've analyzed some of your materials, your gold standard, is it just shows you how much little we know about collagenase, because you could be losing half your activity.
Oh
Uh,
yeah.
and
And
it makes no difference to the cell isolation.
and the thing that yeah, exactly. If it's been left out or whatever, I mean, all these is, and then the other one is, and we learned this from the pancreas, especially the autologous islets in chronic pancreatitis patients. There's such variability in that starting material, and that paper that just came out from Bashu's group,
Right.
where he was looking at specific markers in CP patients and their pancreas, and then determining certain markers that would allow for better isolations than others. We're at the infancy, in fact, as far as doing that. And certainly the FDA is in the greatest infancy, because all fat's the same, it's just structural tissue.
That's right.
No, fat from, I mean, one little thing, and this was pointed out in a publication by Aronowitz, and he said that the Williams has a publication where in dogs, he got 6 to 8 million cells per gram of fat. And I've never answered that in publications. I answered it directly to Joel. That was a series of studies done in dogs from a certain time of year when dogs are known, and these were Texas dogs, to get inflammations that ended up with a huge population of inflammatory cells in their fat. And a majority of those cells, in that 6 to 8 million cells per gram of fat, were inflammatory cells. Now, the SVF was being used for a different application. I didn't need those inflammatory cells, but they did go in to the final activity. But it's little things like that, species differences in fat, and then in the human species, the variability. Do they smoke? Do they drink? What diseases? Do they have inflammatory diseases that end up with lots of inflammatory cells and fat? We're still in the infancy in that. And that's why it's a hell of a lot easier, Kevin, to go and use vesicles, because there's a nice purification method to get those vesicles out of all these different tissues. And then you can characterize them very easy, or allogeneic stem cells. It's just the warning is there might be something special about SVF, that the purified systems, the hierarchical heuristic approach, you may end up with a product that, and Drew, I hate to, I beat up on the quality people a little bit. Process is important, and you saw in there, I said, clinicians, you've got to follow some level of quality systems. Good tissue practices is a good place to start, but focus beyond just the process. Otherwise, you may end up with a door in an airplane that falls out because you're not following the outcomes. The guys who made that door, the quality systems were perfect. They just didn't follow the outcomes. And ask the question, well, you are screwing these things in when you get them, aren't you?
Kevin, you have a question?
Yeah, I just wanted to say I think it was exciting to see an MSC therapy finally get approval in the US, and
Yes.
so maybe that opens the door a little bit wider for the SVF therapies to get regulated either way. I agree with you. By absolute definitions of the 361 wording and language, SVF absolutely meets those requirements, but the FDA's never seen it that way, right? They've always just thought about tissue structural transplant considerations for most of that stuff. So I think these methodological things, using the right collagenases, making sure that you can get rid of those things before you use a secretome-based product, for example, are absolutely critical. So again, this discussion today, I think, was really important to have, and I think we should do more of these in the future.
Yeah,
No,
Kevin, totally agree. The Osiris method, Arnie Kaplan, he was probably applauding the FDA approval. And that was an incredibly important day for all of us. it's approved. I am really pulling that there are no safety issues that come up with that. And it sails through and really helps those patients. That will set the standard. And then we can go and start talking. And maybe FDA will loosen up a little bit and stop reading their 1980 histology book and bring it to current standards of what this is. It's going to make it, for fat, it's going to make it a little bit more complex because we may have to study subcutaneous versus visceral and different forms of fat.
Yep. Well, thanks again. Got to hop, but appreciate it. And
No, thank you.
look forward to the next one.
Good hearing
Thanks,
your voice,
Kevin.
Kevin.
So I'll just end up the session by saying that Drew Brady and I were involved in a call, an editing of the ISO 20399 standard on ancillary materials. And I think one of the things that we're able to contribute, and where we heard from industry reps who had to deal with the ancillary material manufacturers, is the importance of having open communications. And it's also what we're not, how do you say, baffled by, we're realistic, but we had a pharma QA guy come in, and it was unbelievable to him that the people didn't understand the enzymes that they used in their processes. And this was going on all the time. So that's, if you get a chance, that we've written a couple of articles, one of our newsletters, it's on our website, about the importance of the 20399 and the USP standards on ancillary materials. But if we're going to move the field forward, we're going to have to start understanding the characteristics and an optimal dose. I'll give you one example that we did just for the hell of it, is people used Dispase 2 for releasing cells, adherent cells from culture. We found out, and they make a claim that two things, that it's not inhibited by serum. And the second claim is that you can use it for 10 days at 4 degrees. Well, the issue we found out, we know that actually this Dispase, which our version is BP protease, it's GMP purified, is in fact very strongly inhibited by serum. And I think the misnomer is the people using fetal calf serum or fetal bovine serum when they did it. Yeah, it's not fetal. I made that same mistake. Yep, it's fetal serum.
Yeah.
You caught me on that before. Anyways, getting back to it. And the second thing is it loses 60% of its activity within 5 days. So what does this mean? We showed you could use 25 micrograms of Dispase. You don't need, and you're adding all this crap into your system.
Yeah.
Because it's only 10% pure. So again, our plea, as we're discussing a lot of things here, is you can make tremendous improvements in enzymes and really probably cut your costs if you understand your systems and study your systems in relationship to the enzymes.
So, Bob, one of the issues, at least in academics, and it's also in industry, is part of the family jewels were methods.
Yes.
And still are methods.
Yes.
I remember that in the early days, I was in a virology laboratory, and I heard the story of a bacteriophage, I think it was called Phe29. And there was one guy who had developed that, but he wouldn't share it with anybody. So one of his colleagues who was at another university said, well, do you have a letter that the guy wrote saying no, he wouldn't send it to you? And guy said, yeah. And he goes, great, let me have it. And he took the letter and put it into culture. And sure enough, the phage grew out of it.
I've heard this, a similar version of this story, right?
Oh, yeah, because this guy would send these rejection letters out of his laboratory where the phage was everywhere.
Yes.
And that still exists today. I mean,
Yes.
when I am reviewing papers, now for the adipose and the SVF, there are a lot of people who probably say, do not let Stu Williams review this paper or review this grant, because they know me enough now that I'm going to ask for, well, could you provide more details on your method other than, according to our previous methods, and they put a notation in, and I've gone back to that method, and that it has nothing to do with adipose tissue. So what are you doing?
Right.
I don't, maybe it's just gonna be, we're gonna have to start putting up, and I don't know what the incentive is, protocols where everything is listed. Maybe some editors can
What-
give stars out to people.
I would encourage you to go to that Protocols.io webinar.
I have. I mean, there's some.
But they've got everything there. Everything is there.
Yep.
And they, even if you want to do an abbreviated program, you can put it there, but you actually can get it listed as a publication, so you can cite it. And we haven't got to it yet, but as these interns are joining our company, hopefully within the next month, you know, one of the things I'd like to do is just get our enzymatic methods that we use.
Yeah.
That people can see, because that's the question you know, you always get into is, and we don't do anything fancy, and it isn't that we're not going to share it, but you know, you're absolutely right. That's how science was done, and that's how people are ahead of their peers. But I guess what the end result is, we're in a different age now.
Yeah, precisely.
You know, we're translating science into more clinical applications, and there has to be a consensus of what's going on. And you know, in the island world, there's you know, a lot of problems with just people believing other people's results. So
that's right. Now I can read all that German literature just by putting it on the Google Translate.
That's right.
It took me days to do that.
No, I unfortunately, when I got my degree, they waived that, so that was great. They took biometrics instead of biostatistics. Well, anyways, I want to thank everyone for attending this webinar. As I said, if please contact Stuart if you have any questions in more detail, and if any of you have a suggestion of a future webinar, just drop me a note, because I think this is an interesting vehicle where I'm in an interesting channel in which we can have these types of discussions. So again, thank you all, and have a great rest of the day.
Thanks, Bob. Thanks, everyone.
Thank you. Thank you.